In this tutorial, I'm going to first give a brief explanation of what virulence factors are. And in the lab session, I will demonstrate how virulence factors can be identified using a bioinformatics approach. The software that will be used for this task is called Abricate and it's publicly available. I will use the E. coli genome as an example data from which the virulence factors will be identified. Links to the software and genome are given in the description box below, so you can check them out. Now, when we talk about virulence factors, these are mechanisms of factors responsible for microbial pathogenesis. These factors enable the microbes to infect hosts, such as humans. They also cause diseases and other damages to the host. These factors also help the microbes to evade the immune response of the hosts. Examples of virulence factors include adherence factors, invasion factors, and toxins. Now for this tutorial, we will first download an example genome sequence, and then we will install Abricate software using Anaconda. Abricate will now be run on the sequence that has been downloaded to identify the virulence factors. And finally, I'll interpret the results. In order for you to follow this tutorial, you need to first have a Linux environment on your PC because the software runs on Linux environment. Also, you need to have Anaconda installed on the Linux environment you have because Anaconda is what will be used to install the Abricate. I have a video that shows you how to install Anaconda in a Linux environment, so you can check that one out. Link is in the description box, and you can also click the link that's displayed up there for you and watch. So now that you have everything set up, let's begin the analysis, shall we? We will first download the example genome or sequence, and we are going to use the E. coli sakai strain genome. I'll leave a link in the description box so that you can use that to quickly visit this particular page and then download the sequence. In order to download the sequence, you first come to the send to here. Click on this arrow here. Make sure complete record is checked. And when you come to choose destination, click on the file, check it. And then when you come to the format, select fasta. And then when we come to create file, that's what you click. There's the button, so you click on it. And then you'll be asked to save the sequence. You save it in a location that you easily find. I will save it on my desktop. And for the purpose of this tutorial, I will leave it with the default name that has been given. That is sequence.fasta. So I'll save it. So download is complete, so you can quickly check it out before. So this is the sequence that has been downloaded. Okay, and that's the name, and it's on my desktop. And so from here, we will continue the rest of the analysis on the command line. So on the command line, I'll first cd to where I save the sequence. That's desktop. And then I'll just make sure that the sequence is there. So I'll just do an ls and then find that the sequence is there. Let's quickly do some exploration of the sequence. So we first check the header. So there's the first line of the FASTA sequence, and that's the command we are using. You can use that for any text file as well. So it gives us the accession number, the name of the organism, and then some descriptions here as well. Okay, now let's quickly check how many sequences are there. We just want to make sure. Okay, so there's only one sequence. This is a FASTA file. So FASTA files, each sequence begins with this. So having this means there's only one sequence. So that's fine. It's a complete genome, so it's fine. So we have these things intact. So let me just clear the screen first, and then we can proceed. So after we have done this exploration, the next is to add your conda channels. This you can only do if you have installed the Anaconda software package manager. So please do that. So we will add two channels, the Conda Forge channel and the Bioconda channel. So let's begin. We add the Conda Forge first. So we say conda config add channels, and you indicate the channel name, which is conda forge. Let's add a second one, conda config add channels bioconda. So we have the two added. We can confirm by issuing this command, conda config show channels, and that will show the channels for us. So these are the two that we added. These ones, if you don't have it, it's fine. Usually for the default one, it comes when you install conda, so that will always be there. So it's these two that it's important for us at this time. Okay. So after we have the channels added, we will now create an environment and install Abricate. So to create the environment and install Abricate at the same time, we use this command, conda create -n, meaning we are creating a new environment. And we give the environment name. So for this tutorial, we use Abricate as the environment name. And then we give the software, which is also Abricate. So take note of that. And please don't get confused with these two names. So this one is the environment name, and this one is the software that we want to install as well. So let's install them. We hit enter. Please note that this process might take some time. In my case, it took about 10 minutes for the installation to start. So you should have the patience for it. So you'll be asked to confirm the installation. So you type yes. So now installation is complete. So let me just, yeah, so before we proceed, notice that you have this text being displayed here showing that installation is complete. And this instruction tells you how to activate or deactivate that particular environment. So let's clear the screen and continue with the rest of the tutorial. So the next is to activate the environment. But before we even do the activation of the environment, let's confirm that we have the environment created. So we can just say conda env list. It will list all the environments you have on your system. So this one has been created, and that's what we need. So we will now activate that environment. And we do that using the command conda activate Abricate. In older versions, the command is slightly different, so you need to know which version you are using and then use the appropriate command. So we activate with this. So after activation, we will now have this name also appear, and that's the name of the environment. So we can now test the Abricate software. So let's enter some few commands. The first one is to enter just Abricate. Okay, so we have this. So that shows that it's installed. Let's do another one. Let's clear the screen first. Let's just say Abricate list dash list. This will list the databases available. These databases perform different functions, so depending on what you want to do, you check the one that you want to use. I'm going to use the VFDB database. Okay, so that's what we'll do. So what we will do next is to update this database to the latest version, and then we'll use that. And the update is done using the command given here. We say abricate -get db. Then we have this to specify the database we want to update, which in this case is VFDB. And then we issue false. So database will be updated by downloading the latest version. Now we have the update complete. So let's clear the screen first. So it's now time to run the abricate on our sequence. So let's again confirm that we have our sequence there. So we do an ls and it's there. So to run and search for the virulence factors, we issue this command, abricate, and then we specify the database, which is VFDB. Yes, so this is for scanning and identifying the virulence factors. Now we continue again with this command by issuing this .csv, indicating that we want the output formats to be in the CSV format. So that one is also there. We now specify the sequence that we want to find the virulence factors in. And in this case, the sequence name is sequence.fasta. So this is it. Now that we have this command complete, we now save this output to a file. And so we do that by issuing this command, the greater than command here. And then we indicate the file name which the output should be saved in. So we'll just give it a name. We say factors.csv. Let's put it this way. Okay, so we now have it. Or you can also put a meaningful name there as well. So once we have this set, we now run Abricate. Okay, so the task has been completed. So notice that a report is also given here. So it says found 141 genes in sequence.fasta. Okay, so that means we have some hits. Okay, so let's quickly look at the output file. So we do an ls, and this is the file that we specify that the result should be placed into. So we can quickly look at it. So let's just say we want the first 5 lines. Okay, so this is how it appears. Of course, using the command line here, using this command, you have some information, but it's difficult to understand because of how it has been arranged. So of course, you can use some advanced commands to try to um, properly format this information and understand it, but that's beyond the scope of this tutorial. So what we are going to do instead is that we'll open this result, this output file in a spreadsheet software, and then I'll interpret it from there. So let's quickly go and then check it out. So this is the spreadsheet. Okay, so notice that we saved it on the desktop. Okay, that's this particular one here. We saved it on the desktop, and there's the file. So using the spreadsheet software, you open it and it is what you get. Of course, there are several hits. We have 141 as shown in the report. So what I will do is to use an example to interpret the headers that we have here, because this information is very important. So let me switch over to an extract from this result and then interpret it from there. So this is an extract from the results, and we have this information here. So you have the headers which are here, which are these ones here. The ones that I'm highlighting, these are the headers. So each header will have an information, and you need to interpret it, and depending on your questions, you use that to infer. So you have this header here, which is file. This indicates your file name, the file name of the sequence that you input, you ask Abricate to search in. So in this case, ours was sequence.fasta, because this is what we downloaded and saved our genome as. So that is what is given here. So what happens is, if you have several sequences you are scanning from, which are in different files, then these file names will be different for each hit, depending on which of them the hit was discovered in. Let's go to sequence. So sequence here tells you which sequence in that particular file here the hit was identified in. Okay, so if, let's say, this particular file here is a single fasta file. Notice when we did the grep, we got just one hit. When we were initially starting to do the scanning and explore the sequence, we used the grep command, and we found just one hit. So this is the sequence that was in it, and this is where the hit was discovered in. So if in situations where you have several sequences in one file, which is multi-fasta file, then if, let's say, we have sequence A, sequence B, sequence C, and then the hit was discovered in sequence A, then you have that name also being displayed here. If it was sequence B, then you also have that name displayed there here. So this is how this particular information can be interpreted. So it can help you to know the exact sequence in the particular file here that the hit was discovered in. Now when we come to start and end, these are coordinates. These are coordinates in your sequence. The sequence here. Okay, so we have 23330. That's the start. So that means in your sequence, this coordinate here is the start and this is the end. So this range is where the hit was discovered in. So this is how you interpret this particular information. Okay. Now when we come to the strand, you have plus or minus. Okay, indicating forward and the reverse strands. So that's here. So this dash here means that the hit was discovered in this particular strand. Let's go to the gene. So this is the gene name. Okay, that's the virulence factor gene. Let's go to coverage. Coverage here tells us the proportion of gene in the sequence. So this is the gene. Okay, so what proportion of it was found in this particular sequence? And that is what is given here. So we have 1 to 1422, and here is 1422. So it means that this range here is the same as this, meaning that the entire proportion of that particular gene was identified in this sequence. That's what we have here. And this also shows up, we will explain that we have coverage map indicating a visual representation of the hits. So this is the meaning of what you see here. We have aligned, unaligned, and then gaps. Note that during the search of the during the search of the virulence factors, blasting is also performed, and that is why it's important to also visualize what happens with the hits, whether the sequence was aligned or it was unaligned, or there were gaps in it after the blasting stage. Let's go to gaps. Were there gaps in the sequence? Yes or no? If there were gaps, then it's going to be indicated how many of them. We also have percentage coverage, indicates the proportion of gene that is covered. Okay, so we have the coverage here and we have the percentage coverage. So 100% means that the entire gene proportion was covered, and that's also indicated here. Here we had 1 to 1422 out of 1422. So that's what is reflecting here as well. Percentage identity. We are looking at it nucleotide by nucleotide. So the nucleotide in the gene, what did it match the nucleotide in the sequence? That's this one. And that's what we have here. So percentage identity 100. That means that the nucleotides in the gene were the exact matches of that in the sequence that we searched in. That's why we have it 100. Of course, so here we had entire, here we had 100% coverage and identity. So it tells us that the entire gene that was found as a hit. Database indicates what database was used for this task. And of course, we used VFDB. Okay, so that is what is given here. Accession number, accession number also tells us an ID. So using this number here, you can search in some databases to find more information about the hits that you discovered. But that's beyond the scope of this work. You can search in databases like NCBI and others to see if they have the hits, or if this database here, since this database is there, if it's available online, then you can also search that database using this accession number and get more information about your hits. So that's for it. Products, that's a gene product. So this product will give us some additional information. Description. So here we have type 3 secretion system effector, ESP-X1. So this gives us some additional information about the product, what it does. So using this thing, we have an idea of what this particular hit gene does. Now when we come to resistance here, okay, the resistance here is blank. This is probably because of the task we were performing. We were performing virulence factors. But if we were to perform antimicrobial resistance genes, then this particular section would have been filled. But because we are not doing that, that is why it's probably not being filled for us, because it doesn't fit the purpose of this task. So this is just a brief explanation and interpretation of the outputs of the activity that we have just done. But I'll also leave a link in the description box to the developer's webpage so that I can read more about Abricate and then how to interpret your results accordingly. And so this will be all for this tutorial, and then I'll see you in the next session. Goodbye.